Hello, everyone. We are back, and we are going to get into Lecture 2, basically structure, crystalline materials, amorphous materials, even liquid materials, and we're going to talk all about essentially crystalline structure. But we are going to start off, and again, we're going to cover a wide range of materials that have varying ranges of short-range order, which all materials have to some extent, and varying degrees of long-range orientational and long-range translational order. So, do we ever have a perfect crystal? No. Why is that? Well, because even at 0K, but still probably not. But there's always defects in materials. So again, we can look at our delta, got ahead of myself here. In our Gibbs free energy expression, if I have a perfect crystal where everything is kind of arranged as so, and I know where every piece is, and these are all the same atoms, what are the number of ways I can distinguishably configure these? Well, it's just one, and then my entropy would be natural log of 1 microstate, omega equals 1, will be 0. So it's really not feasible that, again, defects will always exist in a material because of that entropy penalty, but we are going to start off, and it's nice to start off with assuming that we are just dealing with a perfect structure. So a crystal structure is simply just a periodic array of atoms. It repeats over long distances. So in a perfect crystal, we are going to know where every single atom is everywhere in space because we can translate that. That structure to infinite space. So we're going to have to define our crystal structure by interaxial angles and by a basis set. So we have long-range periodic order, both translational and orientational. In addition to the short-range order, we're going to have primitive lattices, lattice constants, interaxial angles, symmetries, point groups. Lots of fun stuff to get to. But first, let's just start as simple as we can, which with a 2D lattice. So we can describe that by two lattice constants, A and B, two basis vectors, basically with the magnitude A and B, and one single interaxial angle. So we can look at lots of different symmetry operations, but you can see these two different lattices. So I chose these basis sets. Now you chose a basis set so that you can create a primitive lattice. So what is a primitive lattice? A primitive lattice will basically only have a single atom in its cell. Now, so basically if I kind of connect these with two other basis sets, that's my unit cell. Now you may be saying, well, Professor Steinwell, there's actually 4. You miscounted, you idiot. Not that harsh, but again, let's look at this. Let's look at here. There's 4 atoms, again, technically here, but remember there's another unit cell up top. There's another unit cell right here, another unit cell here, another unit cell down here, another unit cell down here, here, and here. So you can see this atom is shared by what? 1, 2, 3, 4. What about this one? 1, 2, 3, 4. What about here? 1, 2, 3, 4. What about here? 1, 2, 3, 4. So each of these atoms is being shared by 4 other basically primitive cells that exist. So it's not really 4, it's a fourth, a fourth, a fourth, a fourth, which a fourth times 4 is 1. You can see, let's say I said that this was my basis set, here and here. Well, then that would create a cell like this. And then I'd have a cell like this, right next to it, here. You kind of get where this is going. Here, and then here. So now, sure, these are still surrounded by 1, 2, 3, 4. So you have a fourth at the corners, but at the face here, you have half a half. So now, if you chose this unit cell, or this as your primitive cell, it's 2, and it's not primitive. So that's what we have to kind of be careful of when we're choosing that basis set. We don't want to overcount. We need to choose our basis set so that when we create that unit cell, it really is, or that cell, it's a primitive cell. So only one atom. Excellent. There's lots of different types of symmetry and structure. And we're going to get into a lot of these, but we're going to focus primarily on simple cubic, body-centered cubic, face-centered cubic, and we'll touch a little bit on HCP. Lots of different crystals, triclinic, monoclinic, orthorhombic, but again, we are going to focus on cubic crystals because they make up a significant portion of our material, of the materials that we will work with significantly. So these are your friends. Get to know them. We're going to meet them and talk about them a lot. So that's about it for kind of 2D lattices and primitive cells. And next time we're going to get into crystallographic directions. See you then. Thanks.